So we're starting chapter 12, and this is a picture of LOD in an aquarium in the sunlight, and it's creating oxygen. It's photosynthesizing and creating oxygen. And the reason for that is we're going to talk about chemistry in general in very broad terms, but then we're going to talk about redox, and we're going to talk about oxygen being a master element that controls redox, in specifically dissolved oxygen in fresh waters. And this is one of my favorite topics. This is where I figured out I could put ecology and chemistry together and bring all that learning that I had as an undergraduate at about the point you are, maybe, or in maybe graduate school too. Just all that learning that I'd taken in those different classes and put it together and do the things that I loved, since I was a chemistry major and a biology major, and I didn't realize that this whole field of biogeochemistry and how it related to water quality and things that basically keep an environment healthy really was a field of study. So we'll talk about chemicals in general, and then redox potential energy, and chemical transformations. This is going to be a tricky one for y'all. It always is, and so pay attention. Students think they've got it, but they are almost always confused. Then we'll talk about oxygen forms and transformations and photosynthesis. We'll leave the respiration till the next chapter when we're talking about carbon, and we'll talk about how oxygen is distributed in the environment. So the first thing to do is to talk about how we classify chemicals in fresh waters. And you all know the chemical classification by what the elements are and the compounds are, but we have additional things that we're interested in. The first is whether a compound's dissolved or particulate. This is generally a thing of convenience for people that work with water quality. And what it means is what you can filter out versus what you can't. But the fact is that there's some filters that are much finer than others, and so it's sort of an arbitrary level of size, and oftentimes that size is around 0.7 microns or maybe 1.5 microns are the filters that are typically used. And I think we have so we can also think about colloidal versus gravitoidal particles, and that'll be the next slide, so I'll defer that discussion till the next slide. The total dissolved solids, and that's just all the ions that are in the water. And the easy way to do that is to just take a water sample and filter it, so you just have the dissolved stuff, and then you just dry a known volume and weigh what's left, and that's the total dissolved solids. It's related to two other things, salinity and conductivity. Salinity is a relationship of the total salts, and it's related to the sodium chloride content. The more ions there are in solution, the easier it is for electricity to be conducted through that water. So conductivity is a quick measure of salinity or total dissolved solids. And we've used that in the field, where basically all you're doing is you're putting two electrodes down in the water. They're usually a centimeter apart from each other, and then you put a charge across there and see how much of that charge is able to get from one of these to the other, right? And that gives you the resistance, and the conductivity is the opposite of the resistance. Absolutely pure water does not conduct any electricity. So if you're in, we talked about this as well, part of the reason that they kick you out of a swimming pool during a lightning storm, even though you're inside, is that the clouds build charge and then split, right? So there's a really positive or really negative cloud going along in the ground, and the charge is trying to follow that cloud along. It follows along, so there's this charge moving along the ground until it can jump either from the ground up or the cloud down and make lightning, right? But if that charge is going through the ground and it goes through a pool, you're in the pool, and the pool has lots of salts in it, because people pee in the pool and all that kind of stuff. The electricity can be transmitted through that water and zap you while you're in there, right? So that's a graphical illustration of how conductivity might affect you if you're swimming. If you were swimming in an absolute distilled water, you'd be fine. So the next thing is pH, and that's just the negative. It's expressed as the negative log 10 of the hydrogen ion concentration. Really important in determining whether metals are dissolved or not well, and in extreme environments, high pH or low pH. Turbidity is the ability of water to absorb light, and we've talked about that already in the light chapter. And that either can be organic particles such as algae, or it can be inorganic particles such as suspended solids. And so in addition to total dissolved solids, you can have total suspended solids. You can say if you filter something out, weigh what's stuck on that filter, that'll give you suspended solids. And then alkalinity is a measure of basically the bicarbonate equilibrium. And we'll go over that in a good bit more detail when we talk about the bicarbonate equilibrium. So I told you I'd get to this idea of a more natural division of compounds. And one of the real sort of basic cuts is that if you have completely still water, weather stuff will settle out of it. And if it won't settle out, then it's colloidal. And if it will settle out, then it's a particulate. So that actually is a physical thing. The fact, though, is that these colloidal particles, you can never really have a completely still solution, and so it's still a little bit of a fuzzy thing. And then the boundary between colloidal and dissolved is, again, sort of an artificial boundary. So you could have a bunch of cellulose being dissolved, but then if it's in really big chunks, maybe it's becomes glommed up with other stuff like chemic compounds or something like that, and it's not necessarily. So the small ions, hydrogen ion being the smallest one in copper, and then we have some organic materials, small dissolved. Larger things like cellulose and proteins. Chunks of cells are floating around out there. Cells break apart from viruses or something else. And so there's little bits of membranes and stuff and proteins floating around in the environment. And then we get up to the size of a virus. Viruses won't sink. They're small enough that they're colloidal, right? They'll stay in solution. If you have turbulent transport, clays will sink. They're a little bit bigger. And then organisms, the smallest organisms are about this size, and we can get large organic aggregates. And so if you go out into a lake and actually I don't know if you scuba dive or something like that, and you look, you can actually see particles of stuff floating around. And it's just organic aggregates and things like that form. Once you filter the water or put it on through a plankton net, then it kind of breaks that stuff up and you can't see it anymore. But even when you're in the really clean lakes, you'll see these particles, or in the open ocean for that matter, there are these particles around. We can think about, it makes sense that different things are dissolved in different concentrations in fresh waters. And so we have the percent of observations in fresh waters and on the y-axis, and on the x-axis the concentration, that range of concentration. You can see some of these things have ranges over 1,000-fold or more difference. So we can look at some of these compounds that are not very soluble in water, like aluminum, and so the concentration is generally fairly low in natural water. The range of total dissolved solids, the total sum of everything that's in there, can be up to gram per liter. And then some things that are fairly soluble, like sulfate here, right? You can see that sulfate is common and dissolved and really has a wide range of concentrations in it. Something like potassium, this one, right, is a little bit more narrowly constrained. Carbonate is really variable depending upon whether you have limestone or not, right? So, but chloride's fairly constrained, even though the concentrations are high relative to some of these other things. So we'll just keep that in mind, that the natural variation and there's going to be different things that influence the rate that chemicals can be in water. One is their solubility, is a really important one, and that influences not only the concentrations that are achievable in water, but also the ability for them to be weathered off the land surface, which is the source of most of these dissolved compounds in fresh waters. So you can think something like carbonate's high, right? Well, that's limestone. Limestone's quite soluble, right? But manganese and aluminum are not very soluble. Silicate's not very soluble. We talked about that already. So there'd be low constant concentrations. Okay, so here we come to redox. So if you look at this prior graph, you can look, there's all these ions, positive ions, negative ions, different chemistries, and they can take different oxidation reduction states. And the sum of all this sets the amount of free electrons that are able to float around and be available for chemical reactions in waters. And the really key point here is that chemicals have a potential energy if they're at a different redox state than the solution that they are in. So we have here the Gibbs free energy diagrams, and don't panic, don't shut down. This is from chemistry, but this is the potential energy, right? So which has more potential energy, the one that's higher on this diagram or the one that's lower on the diagram? And there's 4 separate diagrams. But on any of the diagrams, if you're further up, do you have more or less potential energy? More, good, Tom. Right, so we can think about this, and you think about this with enzymes, right? You think about this as being a hill. And if you have a ball right here, right, and it has more potential energy than if there's a ball right here. If you want the ball to get over here, you have to push it over the hill. That's the activation energy. So you push it up to the top of the hill, then it drops down. It releases that energy yield, right? So it took this much energy to put it up there, but it yields this total amount of energy and drops down. In this case, we have an oxidizing environment, perhaps one that actually has oxygen dissolved in it, right? So it wants electrons, basically. It's scarfing up electrons. The ammonium has more free electrons than the nitrate. And so if you convert ammonium to nitrate, then you gain potential energy. That means there's a potential source of energy for organisms here. And it turns out this activation energy is pretty high. So if I make a solution of ammonium in water and just let it sit there, it's going to stay ammonium, even though it would have more potential energy, and oxygen's in there. It'd have more potential energy if it converts to nitrate. So what is it that allows organisms to harvest that ammonium to basically make that reaction go? What are the compounds called, and what are they doing? Right, Gary's getting it. Proteins, they have enzymes, they lower the activation energy, right? And what's that called? What's a compound in general called that lowers the activation energy and makes a reaction go more easily?
A catalyst.
Good, yeah, great. Okay, so yeah, proteins are catalysts, and if you are trying to take nitrogen out of the environment and there's only nitrate around and you're in an oxidized environment, and it turns out, we'll find out in a little bit, that you have to turn it into ammonium before you can pull it into your biogeochemical pathways. It is going to take you energy when there's oxygen around. You're going to have to put energy into converting it to ammonium before you can pull it in. Ammonium is NH4, nitrate is NH4 plus, nitrate is NO3 minus, right? So those are the chemical formulas for those ions. A lot of times it'll help you remember if a compound's oxidized if it has a lot of oxygen in it, right? That's how redox is a very general term, but it came about because the first place people figured it out was reactions with oxygen, although it doesn't have to be reactions with oxygen anymore, because oxidations or reductions can happen with other compounds other than oxygen. Now, if we switch this into a reducing environment, let's say we take all the oxygen away, maybe we dump a bunch of sewage into it, right? And the microbes suck all the oxygen down. Then the nitrate has more potential energy than ammonium. And so you're not going to have the ammonium spontaneously going to nitrate. You could have the nitrate spontaneously going to ammonium. And we'll see that there's a biogeochemical pathway at really low oxidation reduction potential where nitrate is converted to ammonium and you can gain energy as an organism from that. Okay, so this is the first time we go through this. What people don't understand is it's relative to the environment that you're in. So I tried to make a schematic of that, right? So the first question is this: there's A and B, right? And we've got these, we can call them molecules. There's blue solid molecules and red cross hatch molecules. In which environment do the blue solid balls have more potential energy, A or B? Just, this will be question of the day if you don't mind typing it in so I get your names down. In which of these, A or B, do the blue solid balls have more potential energy? So everybody's going with A. So why would you argue that it has more potential energy in A? Gabriel's going for B, that's good. Right, so the thing is if you're different from the environment, you're gonna have more potential energy, right? So in this case, the A's, the red crosshatches, have more potential energy in A than B because they're different from most of the things, right? They're more likely to react, find something to react with in A than they are in B, and vice versa. So you could think of these blue things being oxygen molecules, right? And this being maybe reduced iron molecule here. Reduced iron will react with oxygen. And so it has quite a bit of potential energy here. Now reduced iron with very few oxygen molecules around can't find them to react with because the environment that it's in does not have a lot of things that want to take those electrons off of it. So that gives it more potential energy here. Same two compounds, it just depends on what the relative proportions. And you can think about the way it really is in the environment with all the dissolved ions that have more or less desire for electrons. And that's what that redox measurement is, the millivolts of free electrons that are available. And it really tells us what chemical reactions will go spontaneously or at least yield energy, and which it takes organisms to push against potential energy in. And that'll help us understand and organize the entire set of biogeochemical cycles that we'll talk about and why those are important. So we'll keep coming back to this again and again. The fact, though, is that students have trouble with this, so go back and review it. My guess is that your answers are not going to be very well graded on your last quiz. I gave it to you before, so you get it hammered into you. If you didn't understand it, we could go back again. A lot of students do find this somewhat nice because then they struggled through this in chemistry and redox and all that stuff. It's like, well, why does this matter? And then all of a sudden, boom, it matters, because it'll matter if a lake can be cleaned up if the oxidation reduction potential of the sediments is different, for example. Okay, so we can talk now about the oxidation reduction potential of biologically important molecules. And this it's a log scale, just like concentrations of pH, but it's related to the amount of electrons, not the hydrogen ion concentration. So we can start with the first one. If we have a lot of oxygen in the environment, the redox is going to be high. There'll be few electrons available. It's just like pH, it's a flip scale, right? So this means that in contrast, we have oxidation of organic material here. So we can think about, we'll start with an important biological reaction. That is oxygen reacting with organic material. And what do when organisms do this, when they oxidize organic material, what is the name of the process?
I'm just gonna guess combustion.
What?
Combustion?
Combustion is one way to do it. That's true. Although we don't burn, we burn organic carbon, but we don't actually combust it per se. We have another name for it. But it is oxidizing organic carbon and converting it to CO2. Right? What's that metabolic process that does that?
I think Tom put respiration.
Yeah, Tom got it. Respiration, right? So essentially what it's saying is that you change the redox here, you change the redox here, and it turns out you're pushing in oxygen, you're pushing it over the hill. That is, you can get a bunch of energy back from it. So organisms respire and gain a lot of energy. We'll find out in our future, in our nitrogen cycle, that organisms can use nitrate as well and convert it to nitrogen gas and oxidize organic carbon. It's another form of respiration, but you'll see that the redox potential starts a little bit lower and ends up a little bit lower, so the difference gained is less. So they'll only do it when the energy gain is greater than the other reactions. And we're going to go through this again. We'll come back to it, and after we go through all the cycles. But this is what we're going to be talking about time after time. Where an organism might be oxidizing reduced iron to oxidized iron. Why they might want to use manganese. Why ammonia might be converted to nitrate. So we'll keep that in mind. So here's an example of redox and oxygen and iron. And then we're going back to a lake. So we've got a lake that's somewhat eutrophic. Is this lake monomictic or dimictic? So starting at the top, we have temperature, right? And we have warm in the summer here and the surface, and cooler on the bottom, and we've got some ice there. So is it monomictic or dimictic? I know this hurts you that you have to go back and remember stuff that I taught you earlier on in the semester, but that's sort of the point. It all builds. I had a 1. It's dimictic. Cassie got it right. Yeah, it's dimictic, right? So it's not going to be mixing in the winter when the ice is over it, and it's also not mixing when it's stratified in the summer. So the two periods of mixing are here and here. We have a hypolimnion that's setting up. In the winter time, there's not very high respiration, and the oxygen's not going to disappear. But in the summertime, the organic material is accumulating in the top, raining down. The sediment is respiring away the oxygen, and we have an area of 0 oxygen, right? And that's at the bottom of this. This is the hypolimnion during the summer. Now we can look at the redox value when oxygen's around. Remember, I told you that that redox is high, and it's only going to drop low if there's 0 oxygen. So we see it down at the bottom of the sediments, maybe dropping low, but we really only get low redox oxidation reduction potential here in the bottom in the summer when the oxygen's been gone for a long time. So iron can either be in a reduced form or an oxidized form. It precipitates when it's in the oxidized form. And what it does is it converts, it makes ferric hydroxide and precipitates. Do you know what the name of the compound is, ferric hydroxide? What our common name of ferric hydroxide is?
Is it rust?
It is rust, right? So essentially, the water that has no oxygen in it can dissolve iron fairly relatively easily, and you have good high concentrations of total iron coming in and dissolving out of the sediments in here. In contrast to that, when the lake mixes here, right, this iron immediately drops out of solution because it precipitates as rust, as ferric hydroxide. And so you'll have very low concentrations of iron in the rest of the time. I don't know if any of you are on rural drinking water wells that have high iron in them, but what will happen is you'll have an anoxic well and you'll bring the water up and you'll put it into your glass and it will aerate as it goes through that little screen thing, right? And it pushes oxygen into it. And if you let it sit there for half hour, all of a sudden there'll be all this sort of rusty stuff in there that will eventually just flocculent rust that will just settle out. Any of you live in areas that have that in their drinking water? My parents' cabin has that problem. Okay. So there's practical. It won't hurt you. The little iron won't hurt you, right? But it does look kind of yucky. So this will get us into talking about oxygen. About 21% of the atmosphere is oxygen, so that's pretty much. And if there's oxygen present in water, we could call it oxic. And that is in contrast to the older term aerobic versus anaerobic, oxic versus anoxic. Sometimes I might slip into this aerobic, anaerobic. Technically, aero means the atmosphere, right? And oxygen is not all of the atmosphere. What else is in the atmosphere? What's the dominant gas in the atmosphere?
Nitrogen.
Yeah, dinitrogen, right? And what people were meaning when they said something was anaerobic, not that there was no dissolved nitrogen gas in it, they meant there's no dissolved oxygen gas in it. So that's why oxic and anoxic is a more specific terminology. Oxygen is the first thing that drives redox because oxygen loves to react with electrons. And that's oxidation, right? That's the name of where the name of the word comes from. To review, the saturation of oxygen concentration depends on the atmospheric pressure and temperature, and we'll go through that again. And photosynthesis produces oxygen, respiration consumes oxygen. And I'd like you to memorize the equations. So here's a question for you, one that many graduate students don't get right oftentimes, is when a plant is photosynthesizing in the day, is it also respiring?
No.
Doesn't it depend on the plant?
No, it doesn't depend on the plant.
Huh.
What does Emma say?
I think it is always respiring because it always needs to, but it's not always making energy in photosynthesis.
Right, it's not losing energy net, right? Photosynthesis is gaining it energy and it's not respiring as much as it's gaining, but you have to respire. Every living organism has to respire all the time or they're going to die. They can't keep energy going. And this can be an aerobic respiration or an anaerobic respiration, but every organism has to respire all the time. So if you didn't get that right, don't feel bad now because many graduate students have made the same question, failed that same question when I've asked them. However, if you do not get it right, when I do ask you in the quiz, then you can feel bad and perhaps a little bit sheepish, all right? Not terrible, but just, kind of beat up on yourself a little bit so you don't make the same mistake again. Okay, so here's a graph of oxygen, a 3D graph of oxygen saturation concentration. So if you basically have water at some temperature and some altitude, the 100% saturation, the equilibrium will be based on those two factors. As you get water warmer and warmer, right? So zero's here, 50 degrees is here, you have lower and lower saturating oxygen. Okay, that's the first thing. So basically, if you boil water, you can push all the gas off of it. So it's the opposite of many ions where you get higher salt being able to dissolve in water, the hotter the temperature is. But you get less gas being able to stay in water, the higher the temperature is. Altitude also has an effect. So why is there less oxygen in the water at high altitude? Good, Aaron. Less pressure. Lane, less pressure. Right. So basically, less pressure means, right, lower concentration in the atmosphere, because high pressure is high concentration. The number of oxygen molecules per liter is higher in high pressure than it is at low pressure. So as you go up in altitude, there's less oxygen available. If you're trying to hike, you can't get oxygen in as fast because the concentration is low. If you're a fish in the water, the oxygen saturation is going to be lower because the oxygen concentration is lower. So those two factors play off against each other. And this is more specifically barometric pressure is important. It's just that barometric pressure day in and day out doesn't vary that much if you're at one altitude relative to moving, 100,000 meters up or something like that. Okay, so this is important in determining where oxygen is saturating, and it makes a big difference. So let's look at this and say, under what conditions would you expect fish kills to be the most prevalent? First start warm or cold?
Warm.
Warm, right? Because there's less oxygen in the water. And high elevation. So those two kind of go off against each other, right? Because you're more likely to be cold when you're at high elevation. But, if you had a really eutrophic lake at a higher elevation at the same temperature, you're going to have a closer chance of getting fish kills than if you're at lower. Oh, there's another question of the day, which I just answered, basically. You should be able to get that one. Why are more fish kills likely with global warming? Right. And we can let's think about it with a little bit more detail. What's happening to the metabolic rate of the fish with warmer temperatures? Okay, so we've got the metabolic rates speeding up, right? So the fishes their metabolic rate speeding up. So what's happened to their oxygen demand when it goes? They don't explode, right? Oxygen demand that goes up. At the same time, as the water's warmer, there can be less oxygen in it, right? So at the same time as their demand and their metabolism are going up, the oxygen's going down. And so you get this pinch where you tend to have more likely to have fish kills in the summer. Yeah, they don't explode, they go belly up. Well, eventually if they rot enough, maybe they'll, kind of pop, but all right. We'll talk about photosynthetic rate. And we've got a bunch of things that'll come up in the class over time on this. The first point is that net photosynthetic rate is the gross photosynthetic rate minus respiration. So remember I told you that organisms are always respiring. So even when an organism if it's always, a plant is always in the light, it's still got to be respiring it. It can only grow so fast. We'll use this when we talk about in the laboratory using changes in dissolved oxygen to estimate the photosynthetic rate. The net photosynthetic rate is going to be how much carbon is accumulated, right? So how much the plants can grow, whether they're phytoplankton or they're macrophytes or whatever. The gross photosynthetic rate is the total amount of photosynthesis. And there are a lot of different things that can affect photosynthesis. These include light, temperature, water velocity, chemicals like herbicides, for example, that knock. Atrazine, a really abundant, ubiquitous herbicide, works by interfering with the photosynthetic process. That's why it's specific to plants. And we'll talk about the relationships between photosynthesis and light, referred to as photosynthesis irradiance relationships. So we're going to go through that and how organisms adapt. And here we are. So there's a bunch of terminology here that may be new to you unless you've done done plant physiology. And we'll start with this A and talk about the major portions here. We have on the x-axis light going from 0 to 2,000 micromole quanta per meter squared per second. So this would be photosynthetically available or active radiation. And full sunlight is oftentimes around 2,000. Maybe in the tropics can be higher. In the summer here, that's about the maximum we can get. This is the net photosynthesis, right? So it's the balance between respiration and gross photosynthetic rate. At zero light, right, that's just going to be our respiration rate because there's no oxygen production, right? And this is based on oxygen production now, right? So there's no oxygen production. At night, you just respire and that's that. So you basically have a loss of carbon or a negative net photosynthesis if you want to think about that. As the light increases, you get to a point where net photosynthesis is zero. That is, your respiration and your photosynthesis, gross photosynthesis balance each other out. And that's called the compensation point. You're compensating for your respiration, but you're not getting ahead. And then at some point you get up to a maximum photosynthesis, which is called Pmax. So something else has to limit you there. And if the light really goes up high, you can get photoinhibition. You can get basically damage. Ultraviolet can damage your photosynthetic apparatus in different parts of your cell. And you actually start knocking down the photosynthetic rate. This might happen particularly in high altitude lakes, near the surface where the irradiance is super high because the atmosphere is not taking it out, where UV is relatively high. And we'll talk about UV problems because of the loss of stratospheric ozone as we go through this. And the last thing that's in this graph is initial slope. Yes, Emma?
What is this? I've never seen a photosynthesis map quite like this. The light, what exactly is that measured in?
It's measured in micromole quanta per meter squared per second. So it's photosynthetically active radiation.
Is like 400 to 2,000 a strength of light?
Yes, it is. So remember, a mole, how much is a mole in chemistry? Not the little fuzzy thing that crawls underground.
You know that better than I do.
Does anybody have it in their head what a mole is?
6.22 times 10 to the negative 23rd.
Positive 23rd. 6.02 times 10 to the 23rd. Good. Right. So, and that's how we get, molecular weight. So like a gram of hydrogen has 6.02 times 10 to the 23rd hydrogen molecules. Well, quanta can also have our particles, right? So you can measure the number of quanta. So there's a lot of them, right? So then we use this number 10 to the 23rd. You don't want to use that, right? So instead, we use micromoles of quanta coming per each square meter per second.
Aha, so it's particles. I can think of it like that. Like amount of particles coming in to one square. Okay.
Yeah.
I can do that.
Okay. Yeah. Just think of the number of the flux of particles into each square meter, and they have to be between 400 and 700 nanometers because that's the active region of photosynthesis, right? Good. And we have people realizing that chemistry matters. That's good. That they're bringing back their moles of chemistry, and they say they're telling them something. Okay. So the very last thing to talk about here is this initial slope, alpha. It's really important because it gives an idea of how adapted an organism is to low light or high light. And we have these trade-offs between being able to deal with low light and being able to deal with high light. So organisms have photosynthetic units. They have pigments to absorb light. And we've talked about chlorophyll. We've talked about the phycobiolins and the cyanobacteria that absorb green light. They're all sitting on top of that photosynthetic unit trying to get the energy from light to allow for photosynthesis. If you're in really low light, you can synthesize a bunch more of those, right? And capture more of the light. If you're in really high light, evolution says, why waste the trouble, making all those photosynthetic units and those light harvesting when you can't use that much energy? Something else is going to run out. So there's these trade-offs, and we'll see these trade-offs again and again. Nutrient uptake, predation avoidance, all kinds of things. There's ecological trade-offs that evolution acts on. So if you're adapted to low light, you'll have a high or a low compensation point. That is relative, the amount of light it takes you to get to count, is that lower or higher than a high light adapted organism? Lower, good, Cassie. Yeah, right. So that's what it means. You can get to the point where you're breaking even at lower light. So you're adapted to low light, right? That's perfect. But to do that, you're going to have to do a bunch of stuff, pour a bunch of resources into collecting light. And if you do that, you can't necessarily do all the things downstream for photosynthesis, all the other carbon fixing and growing stuff you got to do, right? So you're trading off, and you're not going to have as high of a P max. You'll have a steeper alpha, a lower compensation point, but you're not going to have as high of a P max. And then you really get into a bad situation if you're low light adapted and you move into high light, because you're harvesting all these energetic electrons, and they have nowhere to go because they're not going through the photosynthetic pathway. They are energized molecules that will react with the other molecules in your cell and start ripping things apart, right? And so that's where the inhibition comes in. So low light adapted plants don't do good in high light. Low light adapted algae don't do good in high light. If you have a plant that's a shade plant and you stick it out in the full sun, you can fry it, right? And that's what's going on there. Okay, so this is something that will come up again in the laboratory, and we're right at the end of the lecture. So we'll pick up after this particular graph and the next topic.